Okay, so for this particular discussion, we need to go into the next part of the cardiovascular system, and we need to talk about blood itself. And I'm also going to cover a little bit about the cardiac cycle. And there was one slide in your last presentation that talked about the cardiac cycle a little bit, but I'm going to come back and take that a little bit further. So we're going to begin with the cardiac information. When you look at a cardiac cycle, what we're looking at is what's actually happening within one heartbeat. And typically, if we were in class, I show the typical heartbeat. So you have a little bit of a flat line, and then you're going to get a small little peak. You get a big spike, a little bit of a dip, and then one more little peak, and then another little bit of a flat line. So if you guys think of a typical cardiac reading and what you would recognize as being a heartbeat, that's essentially what we're talking about. But again, it's something that if you see a picture of it, you recognize, oh, that represents a heartbeat. But sometimes there's not a lot of explanation behind what all of those peaks and valleys are actually indicating. So I want to take a couple of minutes and talk through that. Now, as I mentioned, in the cardiac reading, you're going to have one first little spike. It's just a little bit of a peak, and that would be referred to as the P wave. And then you are going to have a very large peak after that, and that is referred to as the QRS wave. And then you have a little bit of a down peak right at the end of your QRS wave, and one little more peak at the very end, and that is the T wave. So it's up, really up, a little bit of down, and then one last up reading, and that is going to indicate one full heartbeat. Now, what's actually happening is in the first little peak that you get, that P wave, you have the atria, which are going to go through a process called depolarization. It sounds really big, but what is actually happening is because the heartbeat is based on electrical impulse, in order to make sure that it only happens when we want it to happen, the cell membranes will keep positive and negative charges separated from each other. So we put the positives on one side of the cell membrane and we put the negatives on the other side. This goes back to our chemistry chapter that we had week 2, semester 1. And so what happens is we use that cell membrane, we separate those positive and those negative charges. And when the time is right, the cell membrane opens up and it allows for those positive and those negatives to interact with each other. Now, anytime that you put a positive and a negative together, they cancel each other out. And that process is called depolarization. So when we talk about that P wave, and the reason that it actually reads as a little blip on the cardiac cycle or the reading is because you have that electricity moving. And it's just enough to cause that little uptick in the monitor. So the P wave is telling you what the atria, the top 2 chambers of the heart, are doing. And then we get to the QRS wave. The QRS wave is going to indicate the activity that is happening within the ventricles. So now we're looking at the bottom 2 chambers of the heart. And when we look at the QRS wave, it is a much higher peak on the reading. And the reason for that, it's the same thing. It's the depolarization of the cells in the ventricles. So we still have those negative and those positives that are interacting with each other and canceling each other out. But it's such a large reading when we get to the ventricles. And this was a feature we talked about in our last discussion. Because the muscle walls around the ventricles are so much thicker than the walls of the atria, when they contract, they do it with a lot more force. And that is going to create the much larger spike on the reading than what we see in the atria. So even though the same thing is happening in the atria and the ventricles during this time period, the readings are so much different in size because of the amount of muscle that's involved. Not as strong of a contraction in the atria, thinner muscles, much stronger in the ventricles because of how thick those muscle walls are. So then we get to the S wave, the S part of the QRS wave, and it will actually dip down below your baseline just a little bit. And the reason for that is this is where you have a change in those negatives and those positives. And because they are moving, you get a little bit of that negative deflection. And that goes back to those negative and positive charges that are involved with this entire heartbeat. The T wave is the last part of this whole process. And what's actually happening, this is called repolarization. And what that means is we talked about the positives and the negatives being separated by that cell membrane. When one heartbeat is over, everybody has to get back into position for the next heartbeat to happen. So all those positives have to go back to where they started, all the negatives have to go back to where they started, and the cell membrane has to close back up. And so all of that repositioning, those positive and those negatives moving, is going to be enough to, again, get a little bit of an uptick on the reading. And that's just telling you that all of those charges are going back to where they need to be in order to cause the next heartbeat in the process. So repolarization, because they are repositioning, they're realigning, going back to where they need to, so the next heartbeat can actually happen. So a little bit of an explanation behind what's actually going on in that heartbeat and what that reading is telling you. Now, in the reading itself, and we could spend an entire semester just on readings and what's going on, but some of the most common ones that you would see in a heartbeat reading would be tachycardia and bradycardia. And in tachycardia, this is going to be where you have a very rapid heartbeat. A lot of times flatline, we don't think of heartbeats and flatlines as being a good thing, but there does need to be a little bit of a flat line between each heartbeat, just so that heart has a chance to kind of regroup, get everything where it needs to be for the next heartbeat to happen. In tachycardia, the heartbeat is happening so quickly that you would still see typically your P waves, your QRS, and all of that, but they're going to happen in such quick succession, you wouldn't see a flat line between them. It would just be heartbeat after heartbeat. In bradycardia, that is a little bit different. That is a slower heartbeat. So you're going to see a longer period of time between the heartbeats, and that's indicating that it's happening at a slower rate than what it typically should. So you can see some of they're referred to as arrhythmias. An arrhythmia will encompass anything in which it's not a typical or common heartbeat. So it could be too fast, too slow. It could be because the atria are doing something they shouldn't be, or the ventricles are doing something off. And that will all give you different readings. And if you were going to go into that branch of medicine, you would get to the point where you could just look at it and know exactly what information it was telling you. Is it something going on with the top of the heart, the bottom of the heart, things of that nature? So the next slide that we have in the packet talks about blood. And from day one, I told you guys that everything in the human body is designed for a particular reason. And we're going to pick up right where we left off with semester one. When we look at the blood cells have a very unique design to them. And when we look at the red blood cells, actually, sorry, I jumped ahead of myself a little bit. Looking at blood itself, blood is considered to be a connective tissue. And the reason that it's connective tissue, and a lot of people don't make the link between why it would be connective tissue. We really don't see, or some people don't understand when you're talking about ligaments and tendons and cartilage, how all of a sudden blood fits into that category. Two reasons why. Number one, blood has what is called a matrix-like design, which means the cells themselves are separated from each other. And that falls into part of the definition of what makes a connective tissue, is having a matrix-like design. The other reason is because in embryological development, the cells that give rise to your cartilage, your tendons, your ligaments, and all of that of the connective tissue group also give rise to your blood cells. So they have the same source of development very early on. And that also puts them into that connective tissue category. So blood, even though we kind of consider it more to be on the liquid side of things, it is considered to be one of our connective tissues. Blood does a lot of things within the body. One of the most important, and we are kind of already talked about this, is that it's going to deliver very vital material to the body. So it takes oxygen to our body, specifically the cells. It's going to take nutrients and other materials that the cells need. It's basically a transportation system. It delivers materials to where they need to go within the body. It also is going to help maintain our body temperature. And it's not the only system that does this. Muscles play a huge role in maintaining our body temp at 98.6 degrees, but the blood is going to help distribute that heat. And so you guys will notice that your body will do different things when your temperature changes. If you start to get cold, you might notice that the blood vessels, especially like in your legs, appear to be purple. That's because those blood vessels are constricting. They're pulling away from the surface of the skin, trying to hold your heat inside your body. And as they pull away from the surface, they tend to appear to be more purple in color. If you get extremely hot, you might notice that your cheeks get red. The blood vessels are now expanding and they are getting closer to the surface of your skin, trying to give off that excess heat. And so there's different things that your body can do with the distribution of that blood, trying to heat up, trying to cool places, so that you can regulate that 98.6 degree body temperature. And then if we were to look at your blood, the main things that we are going to see are the red blood cells, the white blood cells, and even though they're a major part of your blood, white blood cells we're actually not going to talk about until we get into the lymphatic system. They have a huge role in your lymphatic system. And so just to kind of help maintain the amount of information in this chapter, we'll pick those guys up when we get to that chapter. So red blood cells, white blood cells, your platelets, and then your plasma. Now, the next slide talks about your blood volume. And if you look at it, it says that you're as an adult, you are going to carry about 5 liters of blood. And women are going to be 4 to 5, guys are going to be 5 to 6. So that 5 is kind of an average. Children obviously will carry less. And the reason for that difference, women tend to be a little bit shorter, guys tend to be a little bit taller. And so that blood volume will vary just depending on the overall height and size of a person. Now, with the blood, 55% of the blood content is going to be what we call plasma, and it gives you a list of the materials that you would see in the plasma. If you take just the plasma from your blood, it's going to have kind of a yellowish appearance. You can see through it. Some places will describe it as being an amber color, but it's not a clear fluid. There is coloration to the plasma portion. Slightly over half of your blood is going to be that plasma content. And then the remaining 45%, so just under half, is going to be your cell volume. So this is going to be your red blood cells, your white blood cells, and those platelets. The next slide that you have talks about the red blood cells themselves. And when we talk about red blood cells, proper term when we're talking about them is erythrocyte. So you might hear some people use that in place of red blood cell or RBC. They have a very distinct shape to them, and I kind of already started talking about this, but they're not round circles. And when we think about cells, we draw that nice round circle, and red blood cells are a little bit different. They are referred to as being biconcave. And what that actually means is that if you were to look at them from the right point of view, you would notice that it's kind of shaped like a peanut. So they're kind of oblong on the edges, and then they'll dip in through the middle, like somebody took them and kind of pinched them. It doesn't go all the way through the middle. It's not a donut, but it just gets very thin through the middle. And the reason for this is one of the things that your red blood cells has to do is it has to carry oxygen to your cells. And the number of red blood cells that you guys have in your body is massive. You actually lose 1 million red blood cells every single second. 1 million red blood cells every single second are being shed from your body. And in order to have the turnover that we need to replace those without you realizing that they're gone, um, we have a huge number of red blood cells, but they still have trillions and trillions of cells throughout the human body that they have to take care of. And their number one goal is to making sure that every single cell in your body has access to oxygen. So if you're trying to do this massive job of delivering oxygen to trillions of cells, you want to make sure that you are as efficient at it as you possibly can be. The dipped middle portion of the red blood cell is going to increase the amount of space the red blood cell has to hold on to as many oxygen as it can. So it increases the capability of the red blood cell to carry more oxygen than just your typical cell shape if it had that normal round circle that we think of other cells having. Now, the other interesting thing that your red blood cells do in order to maximize how much oxygen it is able to carry is it will start out, typical normal cell has all of the organelles that you would think of your cells having. So we have the nucleus, we have the mitochondria, we have cytoplasm, we have all those other structures, endoplasmic reticulum. And then at a certain point, once it hits maturity, what the red blood cell will do is it will actually kick out the two organelles that take up the most space inside the cell. So the nucleus is going to be the first one. Nucleus is usually the largest organelle, in your cells. Kicks out the nucleus. Now, while that frees up space, it also causes the red blood cell to lose the ability to go through mitosis. Now, again, going back to first semester, mitosis is the process of cell division, the ability of a cell to go from one cell to two new cells. So because it kicks out the nucleus, it loses all of that information that tells it how to go through cell division. So it loses that capability. The other thing is mitochondria. Now, mitochondria themselves are not that large, but the number of mitochondria that you have in some of your cells is very, very high. And so when you put all of those mitochondria together, they take up a lot of space. So they kick out the mitochondria as well. So that is going to increase the amount of available space that the red blood cell has to carry oxygen. After that, the red blood cell is going to actually go through a lot of wear and tear and damage because it's constantly moving in and out of blood vessels. And that wear and tear of forcing itself in and out of these places eventually will cause the red blood cell to become damaged. It breaks down. And around every 120 days, it will end up not being functional anymore and being shed from the body. Now, we don't see this as bleeding from the body. Those dead red blood cells are going to be broken down in a variety of ways, and most of them are going to get sloughed off in our solid waste. So when you go to the bathroom and you eliminate that solid waste from your body, some of the brown coloration of that solid waste is actually coming from the dead red blood cells that are being removed from your system. The other big thing that we have with our red blood cells is something called hemoglobin. Hemoglobin is going to be a protein-based structure, and it is going to basically be where the oxygen are going to hitch on. So it's a special feature that we have in the red blood cell that are able to bind with oxygen so that they can carry the oxygen to your cells and deliver them to your cells. Each hemoglobin can only carry 4 oxygen molecules. So again, that surface area, that extra space that gets freed up by not having the nucleus, by not having those mitochondria, and by having those dips on the surface of the cell will actually provide more room for those hemoglobin, which in turn provides more space for oxygen molecules to be able to bind on, hitch a ride on those red blood cells, and get delivered to your body. Red blood cell counts. The next slide that you guys have takes you through the males, the females, and the children, giving you a little bit of an idea of how many red blood cells you would have in 1 cubic millimeter. So if you look at the male count compared to the female count, it's higher. Now, this is not because they have more blood. We talked about them having 5 to 6 liters, women having 4 to 5. So this is just if you took a cubic millimeter out of a male and compared it to a cubic millimeter of blood from a female, you are going to have more red blood cells in the male than you are in the female. So what does this mean? Going back and kind of linking it together, again, pulling those concepts that we talked about first semester. Oxygen is what we use to make ATP. ATP is the fuel source for our cells to be able to do work. So if red blood cells give oxygen a ride to your cells, and once the oxygen gets there, it is used to make the energy so that your cells can continue to do work. It explains the reason why men are typically going to be faster runners in adulthood. Children and puberty, that's not the case all the time. But once they get to adulthood, it's why if you look at sprinters, if you look at marathon runners, if you look at weightlifters, guys are going to tend to be able to run faster. They have faster paces on sprints and marathons. They lift more. And it's because of that information that you're looking at right there. If they have more red blood cells, that means more oxygen is getting to their cells faster. So as they're doing these activities, as they're running, as they're lifting, the oxygen getting delivered to their cells happens at a faster, higher rate. They are able to produce and make AT faster and get that fuel source replenished to their cells at a faster rate than females. So the turnover rate on that is quicker. So their cells can work a little bit longer, a little bit harder than what you can in other genders. And that's not a slam on the female. It is just a physiological explanation as to why guys tend to, you see those differences. All right, if you go to the other components of the blood, so in the